Hello everyone, and welcome to episode 16, Remastered. In the last few episodes, I've been discussing DNA. I've been talking about chromosomes and genes and alleles. In the last episode, I specifically talked about transcription and translation, which was dotted with a few explanations here and there on the structure of various parts of the DNA molecule. In today's episode, I'm going to be discussing how DNA is produced, how it's generated or synthesized, and how it's repaired. Naturally, any discussion about DNA repair has to involve at least a little talk about DNA damage, specifically mutations. I'll end this episode by talking about the different kinds of mutations and what they mean for the organism whose DNA has suffered from that particular kind of mutation. So with that said, let's dive right into it. Deoxyribonucleic acid is composed of two strands, wrapped together in a helix. Each strand in the helix is complementary to the other. This is to say that they're each lined with a series of nucleotide bases, A, T, C, or G, and these bases interlock in pairs to create complementary strands. The A and T bases, or adenine and thymine, are able to pair together. Similarly, the C and G bases, or cytosine and guanine, also pair together. When one strand has a sequence like ATCCG, the other strand has a complementary sequence that goes TAGGC. This is called complementary base pairing, and it's really important. Recall the directionality of DNA that I described in episode 4. The backbone of DNA is composed of ribose sugars and phosphate groups, all linked together in this repeating pattern. The phosphate groups will bind to the 5' carbon of one sugar and the 3' carbon of the next sugar. As the ribose sugar polymer forms a strand, it'll have a 3' end, where a hydroxyl group on the 3' carbon is exposed, and on the other end it'll be the 5' end, where a phosphate group dangles off the end of the 5' carbon of the last sugar. I should also mention that DNA is anti-parallel. This means that the DNA strands run in opposite directions from one another. They're parallel, but they just run in different directions. If you were to take a molecule of DNA and lay it down in a straight line with one end on your left and one end on your right, each end of the DNA molecule would have both a 5' and a 3' end, one from each of the strands. If you were to split the DNA into its separate strands, well, one strand would have its 3' end by your left and its 5' end by your right, while the other strand would be oriented in the opposite direction. During DNA synthesis, new nucleotide bases are only added to the 3' end. This makes some aspects of DNA replication relatively complicated. Now, I talked about DNA replication a little bit during the S phase of the cell cycle, but now I'm going to go into much more detail. DNA displays a semi-conservative replication. This means that during replication, one molecule of DNA is split into its two component strands. Each strand will then serve as a template for the creation of a complementary strand. When synthesis of the complementary strands is complete, we now have two new molecules of identical DNA. The DNA has been replicated. And this is semi-conservative, because each molecule of daughter DNA has one strand from the original parent DNA molecule, in addition to their new strand that was recently synthesized. So one strand of DNA gets preserved from one parent generation to the next, and the new strand is synthesized complementary to that. The actual process of DNA replication involves a large suite of enzymes and protein complexes. The enzyme that actually adds new nucleotide bases to a newly forming strand is called DNA polymerase. Nucleotide bases are only added on the 3' end because that's the only way that DNA polymerase can attach new bases. It's the structure of the DNA polymerase that causes this limitation. The enzyme can only interact with the reactive hydroxyl group on the 3' end. As the DNA polymerase does its job, it slides down the newly forming DNA strand in a 5' to 3' direction. These new bases are added to the strain through phosphorylation reactions by molecules called DNTPs. Recall the molecule ATP. It stands for adenosine triphosphate. Now, you might have noticed that adenosine is also a nucleotide base. If you listened to earlier episodes, you also might have heard me mention GTP, or guanine triphosphate. This is a similar energy-carrying molecule, but it just uses the nucleotide guanine instead of adenine. Now, cytosine and thymine can also be used in this energy-carrying molecule format. Now, when a nucleotide base from DNA is bound to a triphosphate group, it becomes DNTP. Can you guess what DNTP stands for? It means Deoxyribonucleic triphosphate, because it uses nucleotides derived from and used for deoxyribonucleic acid. It's the whole base. It's not just the guanine or the adenine molecule. It's the sugar and the phosphate group as well. Okay, so now that you know the enzyme that adds nucleotides, the direction that it works in, and a little bit about the DNTPs that it uses as building materials, let's get into the mechanisms of replication itself. The process of replicating a molecule of DNA begins at a place called the origin of replication. Yeah, you might have noticed a trend with the really creative names that biologists use. Anyway, this origin of replication is it's a point in the DNA strand where it first starts to split apart. Bacteria only have one origin of replication in their DNA, but eukaryotes have a lot of different origins of replication. When the DNA strand splits apart at the origin of replication, a suite of proteins facilitates the split and allows it to continue. The nucleotide bases in both directions from the origin begin to unzip. A bubble emerges, a bubble that's formed by the growing space between the two strands. At each corner of this almond-shaped bubble, the DNA helix is being split apart into two separate strands. Now, these corner points where the DNA helix is being actively unwrapped are called replication forks, as they have a Y shape that resembles a fork in the road. So the first of these proteins that comes in and swarms the origin of replication is DNA helicase. DNA helicase will bind to the origin and break the hydrogen bonds between the nucleotide bases. This breaking of the hydrogen bonds initiates the breakage of the two strands, and it begins the formation of the replication bubble. A group of proteins called single-strand DNA binding proteins, or SSBPs, will come along to help the DNA helicase do its job. This is because if the DNA strands are cleaved apart and then just ignored, they'll rapidly reattach, they'll just recombine, and if this happens, then the DNA helicase will not be doing anything productive. It'll just be running down the length of the DNA, unzipping the bases, only for the DNA to naturally zip back up right behind it. The SSBPs prevent this from happening. As their name suggests, the SSBPs will bind to one of the two separated DNA strands and keep them from reassociating with each other. This keeps the replication fork open so that the bubble can continue to expand. As the DNA helicase works its way down a strand of DNA, unzipping the bases at the replication fork, it creates a tension in the unzipped portion of the DNA. If you have a string of yarn, you'll notice that the string of yarn is made of smaller strings all wound together. If you grab the tips of those small strings and pull, in an attempt to unravel the strand of yarn, you'll quickly cause the strand to coil up with tension. DNA is not much different. As the DNA helicase unzips the DNA and the SSBPs pull each strand apart, tension is induced in the unzipped portion of the DNA. An enzyme called topoisomerase will move ahead of the DNA helicase, and it'll make cuts in the DNA to relieve this tension, and it'll reseal those cuts before the DNA helicase gets to it. In this way, the topoisomerase is kind of like a pressure gauge that lets out some steam when the pressure gets too high. When the DNA gets too tightly wound up, the topoisomerase just makes a few snips and cuts, it'll relieve the tension, and then it'll patch everything back up and move along. The topoisomerase will make a few snips and cuts, it'll relieve the tension, and then it'll patch everything back up and move along. So now that the DNA is opened up, it's time to start synthesizing the new strand. Recall that DNA is only synthesized in the 3' to 5' direction, as DNA polymerase can only add bases to the 3' end. DNA polymerase also needs a base with an exposed 3' end to start off of. It can't just come in and start laying down its own tracks, so to speak. It needs a starting point. So where does the DNA polymerase start if the replication bubble only has 2 single-sided strands of DNA? Well, it starts with a small piece of RNA called a primer. This primer is small, typically only 3 to 15 bases long, give or take. An enzyme called RNA polymerase will find the origin of replication and bind to it. Now, as the replication bubble grows and the replication forks spread out farther and farther down each side of the DNA strand, the original origin of replication will stay right where it was. It'll stay in the middle of each single strand, where the replication bubble began. So this RNA polymerase will go out to the middle of each free-floating strand and associate with that starting sequence. Unlike DNA polymerase, RNA polymerase doesn't need a 3' end to start synthesizing its product. So the RNA polymerase will bind to the origin of replication after the replication bubble has already started forming, and it'll build a short strand of RNA, called the primer, right at the origin of replication. It'll be complementary to the origin of replication on the DNA. This RNA primer provides the 3' end that the DNA polymerase needs to start synthesizing new DNA. So the RNA polymerase comes in, binds to the origin of replication, makes this little RNA primer, and that is like a groundwork for the DNA polymerase to work off of. So the DNA polymerase will come in, bind to that RNA primer, and then start building off of that. It'll start synthesizing new DNA by moving in the 5' to 3' direction. Now, this newly created strand of DNA is called the leading strand, as its synthesis is smooth and continuous. As more nucleotide bases are added to the 3' end, the 3' end will approach the replication fork. But the replication fork keeps moving downstream as the bases are unzipped by DNA helicase. So the 3' end of the newly synthesized strand won't ever really catch up to the replication fork, so the leading strand can be synthesized continuously in a very direct and very simple manner. But remember that DNA is anti-parallel. So as the DNA polymerase is building this new strand of DNA in a 5' to 3' direction, as it's going along and doing this, it's running along the template strand in the 3' to 5' direction. So you can see that anti-parallel quality there. Now you gotta consider the anti-parallel quality in the other strand that was originally... Let me try and describe this in a way that will allow you to visualize it. So let's say that you're sitting on top of the DNA helicase molecule as it's unzipping the DNA. If you look forward, you'll see the double strand of DNA extending before you like train tracks. You'll see the topoisomerase busying itself around in front of you, cutting and releasing tension in the DNA. But if you're looking in front of you, this is really all you're going to see. But if you turn around to look behind you, like you were in a car and you looked out of the rear window to see the road that you'd already covered disappear behind you, well then you would see this expanding replication bubble, and you would see all of these proteins and enzymes and stuff swimming all around it doing the DNA synthesis. You would see that the replication fork exists right underneath you, and as the DNA helix is getting unzipped, one strand is branching away to your right, and another strand is branching away to your left. To your left, you see the DNA polymerase of the leading strand. The DNA polymerase on the leading strand will be chugging along doing its thing right behind you on the DNA helicase. It's building that new strand of DNA, the leading strand, in one single continuous effort. But now look to your right. What would you see over here? Because DNA is anti-parallel, this other strand runs in the opposite direction from the strand on the left. This means that DNA polymerase can't chug along right behind you. As it can only add nucleotide bases to the 3' end, it'll be moving away from you in the DNA helicase as it synthesizes its new strand. This new strand on this side is called the lagging strand, or the discontinuous strand, because it has to be synthesized in discrete chunks. Basically, RNA polymerase has to come and make a new primer at some point along the strand. DNA polymerase will come and bind to the primer and begin synthesis in the 5' to 3' direction. Eventually, the DNA polymerase will come to the middle of the replication bubble, to the origin of replication, where it runs into the leading strand going in the other direction. The DNA polymerase will detach after having synthesized a small stretch of new DNA. The RNA polymerase will then go off to a point further down the strand to make a new primer, and the process will repeat itself. The RNA polymerase makes a primer, the DNA polymerase associates with that primer, and then makes a short stretch of new DNA, and then stops when it bumps into the end of the previous DNA segment that it just made. The DNA on the lagging strand is essentially synthesized in short segments that connect together. These segments are called Okazaki fragments, named after the researcher whose team discovered them. RNA polymerase will then make a new primer, and DNA polymerase will make a new segment. This process will repeat over and over again in these discrete steps, building the lagging DNA strand one Okazaki fragment at a time. An enzyme called DNA ligase will come through and seal all of the little breaks between the Okazaki fragments, smoothing them out into a continuous strand of new DNA. So let's go back to that metaphor where you're riding on the DNA helicase and looking back at the replication bubble as it expands. To your left, you see the DNA polymerase of the leading strand following close behind you. To your right, you see RNA and DNA polymerase doing this kind of back and forth shuffle as they synthesize the Okazaki fragments. DNA ligase lingers behind them both, smoothing out the fragment sections into a single continuous strand of new DNA. Now eventually, as the DNA helicase runs towards each end of the DNA molecule and the replication bubble expands, eventually the two original strands of the DNA will be completely split apart. And thanks to the RNA and DNA polymerase, a new complementary DNA strand is being built along each of the parent strands, and when they're done, you'll have two new identical DNA molecules. This is how DNA is replicated in a semi-conservative fashion. So this sounds pretty easy and straightforward, right? I mean, open up the DNA at the origin, make the replication bubble, build your leading and lagging strands, and you're set, right? Not quite. You see, DNA polymerase requires a primer to start synthesis, as I said. When a primer for an Okazaki fragment is not needed anymore, as in the DNA polymerase has already launched from it, then an enzyme will come and eat it away. It'll eat the primer. The next Okazaki fragment will go right up to the start of the previous fragment. In this way, the new DNA molecule is purely DNA. It's not peppered with these little chunks of RNA left over from the primers. When the primer is on the end of a DNA molecule, it'll be decayed away. But when this happens, there's no place for a new primer to attach. DNA polymerase will be unable to fill in the space where the primer used to be. Where each leading strand is a full replication of the template strand, each lagging strand replicates the template strand with the exception of this last little tail end of the DNA where the primer used to be, as well as any stretch of DNA that dangles past it. That small stretch of DNA on the end of the template strand can't be replicated. Now you might be thinking, what's the big deal? Who cares? It's just a few nucleotide bases. Well, technically it's about 50 to 100 bases. But the DNA molecule is billions of bases long. How can a loss of 100 bases among billions matter? The problem is that this tiny little loss of information happens every single time the DNA gets replicated. Every single time the DNA is replicated, every single time a cell replicates itself, a small bit on the end doesn't get replicated, and the strand of DNA shrinks. When this strand is itself replicated, its daughter strand will be even shorter, and that one's daughter strand shorter still. As time goes on, the DNA continues to replicate over and over and over again. This little bit will add up, and soon the DNA molecule is suffering serious degradation at its tips. And when your DNA degrades, your cells and your body degrade. Our bodies prevent this genetic degradation with structures called telomeres. If a strand of DNA is like a shoelace, the telomeres are like the aglet, keeping the shoelace from fraying and falling apart. Telomeres are composed of repeating sequences of regular DNA, but they just don't code for anything. They're not genes. In humans, for example, the telomeres are composed of thousands and thousands of repetitions of the sequence TTAGGG. An enzyme called telomerase is responsible for building telomeres. Telomerase is the kind of enzyme that isn't strictly a protein. Like the ribosome, the telomerase enzyme also contains a little bit of RNA. The telomerase uses its RNA component to bind to the bases at the end of the template strand. From here, the telomerase will add bases to the template strand, not to the new strand, and this will cause the template strand to extend a little bit. And by extending the telomere, it creates another site for the RNA polymerase to create a primer. It's important to understand that this process with telomerase doesn't actually stop the little bit of DNA on the very end from not getting replicated. That still happens. It's just that with the help of telomerase, the template strand can get expanded well past the limit of any actual genes. When the lagging strand is synthesized, the little bit at the end that doesn't get replicated is just part of the telomere. It isn't part of any important gene or protein coding region. If those more important regions got damaged, then their function could be impaired, and that would have health consequences for the organism. In humans, telomerase is only active in the cells that produce gametes. That is, the telomerase only actively works to rebuild and rejuvenate our telomeres in the cells that produce sperm and eggs. Every other cell in our bodies, like our skin cells, or our lung cells, or our brain cells, these are called somatic cells. Unlike the germ cells that make sperm and eggs, somatic cells do not undergo meiosis. They only go through mitosis. And on that note, somatic cells only go through mitosis a limited number of times. As somatic cells don't possess telomerase, their telomeres get degraded and don't get repaired. Once the telomeres have been fully chewed up and destroyed, the DNA itself will start to be degraded. Instead of continually replicating DNA that just gets worse and worse quality, the cell will reach a point called senescence, where it simply stops dividing entirely. The cell basically decides it can no longer pass on its sufficiently degraded DNA, and it'll just stop reproducing. One of the leading theories on aging posits that we age because of our cells going into senescence. When you're a newborn infant, your cells haven't had time to replicate that much, and they're very far from senescence. But as you age, your cells will start to go into senescence more and more. It's slow at first, not even noticeable compared to the rate of cells still dividing and making new, healthy daughter cells, but as time goes on, more and more and more of your cells will slip into senescence, until they become a non-trivial portion of all of your tissues. You'll find that your skin will start to wrinkle and droop as the cells who once maintained the collagen are going into retirement. You begin to get forgetful and confused as brain cells go into senescence and shrivel away. Your vision fades as cells in your eyes follow the same fate. And so it goes throughout your whole body, until so many cells are in senescence that you reach a critical point where your cells simply aren't dividing in the numbers necessary to sustain your life, and you die. Naturally, research into cures or treatments for aging have put intense focus on telomeres and telomerase, specifically into finding a way to activate telomerase in our somatic cells, and not just in the germ cells that produce gametes. Now, there is a circumstance where somatic cells do express telomerase. This is called cancer. Cancerous somatic cells don't have proper regulatory control over their own replication. And because of this, most cancer cells are actually able to express the genes that produce telomerase. Cancer cells can thus protect and maintain their telomeres, while your healthy cells suffer from the ravages of time and age. For a cell to become cancerous, it has to have some kind of error or mutation somewhere in its DNA that disables the regulatory mechanisms involved in cellular replication. Not all mutations cause cancers, but they can impair other functions of the cell. Furthermore, not all errors in the DNA are due to mutation. Some errors, for example, are from mistakes that are made during the process of DNA replication. Despite working at rates of up to 500 bases a second, DNA polymerase rarely makes mistakes. In fact, less than 1 in 100,000 nucleotide bases organized by DNA polymerase fail to match its associated base on the template strand. How can DNA replication be so precise? And when this replication fails and creates an error, how is it fixed? So first, understand that nucleotide bases don't pair up the way they do for no reason. Adenine and thymine pair together, and cytosine and guanine pair together because these molecules have structures that fit together really well, and the conformations that they make are the most stable. This means that these pairs of nucleotide bases are in their most energetically favorable conformations when they're paired with their complementary base. When synthesizing a new strand, DNA polymerase is unlikely to make a mismatched base pair, as that would mean doing something that wasn't energetically favorable. It would be a very noticeable bump in the road from the DNA polymerase's perspective. If the DNA polymerase encounters one of these bumps in the road, one of these mismatched base pairs, it will immediately halt everything in order to remove the offending nucleotide base. After removing the mismatched base, the DNA polymerase continues to synthesize more DNA. Despite its best efforts, the DNA polymerase can still skip over a mismatched pair from time to time. And when this happens, a suite of proteins will jump into play to fix the overlooked mismatch. As DNA polymerase makes about one mistake for every 100,000 bases, the assistance of these proteins reduces the error rate for DNA down to about 1 in a billion bases. DNA doesn't just suffer problems from mistakes that are made during replication and proofreading. DNA can also be harmed and mutated by all sorts of environmental inputs, like cigarette smoke, ultraviolet radiation, and industrial chemicals like Agent Orange. Typically, our bodies employ various enzymes that work through various mechanisms to repair this daily damage. Otherwise, the mutations would build up rapidly and kill us quicker than senescence. One such mechanism for DNA repair is called nucleotide excision. When ultraviolet radiation strikes a molecule of DNA, its energy can cause two adjacent pyrimidines to bind together into a dimer. So if UV radiation hits two adjacent cytosines, it can fuse them together to make a cytosine dimer. Similarly, if UV radiation hits two adjacent thymines, it can fuse them together to make a thymine dimer. These dimers are like awkward kinks in the DNA that screw up its structure and impair the function of enzymes and proteins that are supposed to slide along the DNA molecule. To mitigate this damage, the cell determines which segment of the two DNA strands of the DNA has been damaged. The segment of that damaged strand is then removed and destroyed, and it gets replaced by DNA built off of the template of the remaining strand. So at this point, I've mentioned mutations quite a few times, especially in the context of cancer cells, but I haven't yet gotten around to explaining what mutations are. To put it in a very technical sense, a mutation is a permanent alteration to an organism's DNA that gets passed on to its offspring. In this way, mutations are much more than the mundane ultraviolet damage that gets inflicted and repaired in a day's time. Mutations are permanent alterations to the genome, which leads to the creation of new alleles that code for new traits. Unfortunately, the vast majority of mutations are harmful, and the so-called new alleles that they make code for something like a less effective lung tissue, or a malformed eye, or it leads to mental retardation. Mutations can affect a single nucleotide base, or they can affect multiple bases. Those mutations that affect a single base are called point mutations. Let's say that we have a gene with the sequence TCTCAG. This would produce an mRNA molecule with the sequence AGAGUC. Now, real genes are hundreds, if not thousands, of bases long, but this is just a simple example, so bear with me. Let's say that the third base in this little tiny mRNA molecule, an adenosine, is removed in a point mutation and replaced by a cytosine. Now the mRNA sequence goes AGCGUC. If you listened to the previous episode about transcription and translation, you know that groups of 3 sequential nucleotide bases code for specific amino acids. And these amino acids get added together to a polypeptide chain that eventually becomes a protein. The type of protein is determined by its amino acid sequences, and the amino acids are determined by the 3 bases, or the codon, that codes for it. I discussed how most amino acids have 2 or 3 different codons that code for them. This is kind of like a built-in redundancy to protect against mutations. So let's go back to our gene sequence for a moment. The original sequence on the mRNA molecule was AGAGUC, but the point mutation turned it into AGCGUC. If AGA is our first original codon, and GUC is our second original codon, we can see that our first codon has suffered the mutation to its third base. The first codon, AGA, codes for the amino acid arginine, and the second codon, GUC, codes for valine. But now that the AGA codon has become mutated to AGU, it doesn't code for arginine anymore. It codes for an amino acid called serine. The original gene would have coded for a polypeptide composed of arginine and valine monomers, but the mutated gene now codes for a polypeptide with serine and valine. The protein is new, it's a product of mutation. But in most cases, like 99.9999% of cases, this mutation produces a non-functional protein. Or worse, it'll create an actively harmful protein. But in those extremely rare cases, in those extremely rare 0.0000001% of instances, I'm kind of exaggerating, but you get the idea. In these extremely rare cases, the mutation produces a protein that works better than the original, or it provides a novel function that the original protein doesn't have. If this novel function provides a fitness advantage for the organism, it'll be more likely to survive and reproduce, passing on this mutant gene to its offspring. If the mutation increases the parent's fitness, it will likely increase the fitness of the offspring as well. In this way, the mutation will persist in the gene pool, spreading in popularity as it outcompetes and outbreeds those individuals who possess the inferior original allele. In that example with the hypothetical gene sequence, the point mutation in question is also called a missense mutation. A missense mutation is one that ultimately causes a change to the amino acid sequence in a protein. Then you have silent mutations. A silent mutation is a mutation that doesn't change the amino acid of a protein. Okay, so let me think of an example of this. Consider the codons GGU and GGC. Both of these codons code for an amino acid called glycine. If a GGU sequence were to undergo a point mutation to become GGC, this would be a silent mutation, as it didn't change the encoded amino acid. If the encoded amino acid isn't changed, the protein isn't changed. If the protein isn't changed, there's no problem. The silent mutation doesn't cause problems. It's silent. There's other kinds of mutations, and these don't involve changing one base into another. Some mutations involve the wholesale addition or subtraction of bases from a gene. The addition or subtraction of a single nucleotide base can ruin an entire protein. As the bases in the genetic code are read in groups of 3, adding or subtracting a single base will shift the entire frame of reference and totally scramble the coded message. These kinds of mutations are called frameshift mutations because they literally shift the frame. Let's use our original gene sequence again to illustrate this, the AGAGUC sequence. So imagine that a cytosine has been shoved into our sequence. There's a frameshift mutation where a cytosine base has been added in. It's been added between, let's say, the first A and G, so that now the sequence reads ACGAGUC. Our original two codons coded for arginine and valine, but those codons don't exist anymore. The new codon is ACG, and the next codon is AGU, and these code for the amino acids threonine and serine respectively. The frame has been shifted down by one base, and as a result, the last base in the sequence, that last cytosine there on the end, is all by itself. Because it's just one base, not three, it's not part of a codon, so it can't code for anything. It's just sitting there on the end of the DNA, wasted. A second kind of mutation that radically changes the encoded protein is a nonsense mutation. Where a missense mutation creates a protein with a different amino acid sequence, a nonsense mutation creates an artificially short protein. You see, there are three codons that are called stop codons, because when they go through the molecular machinery involved in translation, they dislodge the polypeptide and shut everything down. This stops the ribosomes from adding more amino acids, and it creates a free-floating polypeptide chain, which can then fold into a protein. A nonsense mutation happens when an otherwise normal codon is mutated into a stop codon. Where the normal polypeptide should have another amino acid added on, the nonsense mutation causes the protein to be dislodged prematurely. As the protein hasn't been fully constructed, it can't fold properly. The affected protein is basically useless. Now, mutations aren't limited to the DNA sequence itself. The chromosomes can also suffer from mutations. In previous episodes, I've discussed polyploidy, where you have an inappropriate number of copies of a chromosome, and how this can lead to serious medical complications like Down syndrome or XXX syndrome. But simply separating the chromosomes incorrectly is not the only way that chromosomes can be mutated. Segments of chromosomes can be organized or replicated improperly, leading to the impairment of the total chromosome. Sometimes a segment of a chromosome can be totally lost in a mass deletion event. Other times, a segment of a chromosome can be copied. There's more copies than the chromosome needs, and this is called a duplication event. Recall that during meiosis, chromosomes are deliberately broken at specific points before a crossing over event. Sometimes, accidental breaks in random places can occur, and instead of being cleanly repaired, these broken segments may flip and reattach with the wrong ends. This flip is called a chromosome inversion. In more extreme cases, the broken segments won't even reattach to the other segment of their own chromosome. Instead, they'll reattach to segments of an entirely different chromosome in an event called Chromosome Translocation. Here's a really interesting fact about this. Millions of years ago, there was a primate ancestor of humans that experienced a chromosome translocation. The lineage that produced this chromosome translocation would go on to produce humans. But the lineage that didn't have this chromosome translocation would diverge and produce the genus Pan, which would give rise to chimps and bonobos. So one of the big genetic events that caused humans to speciate from chimps and bonobos was a massive chromosome translocation. I think I've covered all the major points. I hope you were able to learn something cool from this episode. And as always, thanks for listening.